How is the enhanced ability to detect changes at a cued location, which we looked at in the last video, reflected in brain activity? First, let's look at what happened to the firing rate of visually driven neurons in response to the oriented gratings in the same orientation change detection task that we just discussed. In this study, they were performing extracellular recordings from an area of the brain called V4, where neurons respond to visual stimuli. When they were finding a neuron to record from and setting the task up each day, they would make sure that one of the stimuli being presented on the screen appeared in the receptive field of the neuron being recorded. The other grating is in the opposite side of the visual field and is being represented primarily by neurons in the opposite hemisphere of the brain. So the appearance of the grating in the neuron's RF causes this neuron to increase its firing rate. To see if this neuron is affected by where the animal's attention is directed, we want to compare this firing rate when the grating appearing in the RF was cued versus when the other grating was cued. Here's a plot of the neuron's firing rate after the grating appears in its RF over time. When the grating was attended, black, versus not attended, shown in gray. You can see that the firing rate is a little bit higher when the grating in the neuron's RF was attended. This fits intuitively with our understanding of how brain activity relates to perception. Higher firing rates mean a stronger representation of the attended stimulus, which means we're more likely to notice a change. This has now been shown in multiple visual areas, all the way from V1, primary visual cortex, up to inferotemporal cortex, one of the final stages of object processing in the brain. As it turns out, this simple firing rate measurement isn't the only way that the brain's representation of a visual stimulus changes during attention. The reliability of firing rates between different trials increases, the chance of two neurons firing at the same time changes, and the timing of spikes relative to those of other nearby neurons also changes. But the firing rate effect was the first to be measured and one of the easiest to think about. So these changes in the responses in brain areas that process visual stimuli could be the basis for enhanced perception of those stimuli. But what causes these changes? It seems like the source of these changes must be non-visual areas for a couple of reasons. Firstly, we don't see firing rate changes in visual areas when we're waiting for a stimulus to appear, but that cue information must be held somewhere in the brain. Second, we can also use non-visual cues to direct attention. Those are auditory cues, for example. So, where do we see changes in sustained activity that reflect the cued location for attention? In certain areas within prefrontal and parietal cortex, we see activity that looks promising as a source of attentional modulation, as in this example, looking at the average activity of many neurons within a prefrontal area called the frontal eye field. Neurons here respond to the attentional cue, and like visual areas, they show enhanced responses to visual stimuli at the cued location. Unlike purely visual areas, they also show sustained activity at the attended location in between the cue and the visual stimulus, and again in between the two stimuli. Human fMRI data has likewise implicated a network of prefrontal and parietal areas as active during attention. Many of these areas are also involved in the control of eye movements. Areas in prefrontal and parietal cortex that display this type of sustained activity at the location of attention are good candidates as the sources of attentional modulation in sensory areas. To really test this idea, you want to do more than just measure activity in these areas. You want to alter activity in these potential source areas and determine whether that changes sensory processing. These methods for manipulating neural activity will be the subject of the next video.